student, welcome to chemistry class. Students, in this lecture we will discuss about the differences between solution, colloids and suspension. So we have already studied about the definition, what is solution, what are colloids and what is suspension. Now let's see how all the three are different with each other. Now, as we all know, solution is a homogeneous mixture, right? But colloids and suspension, they are categorized under heterogeneous mixture, right? And you all know homogeneous and heterogeneous mixture. Homogeneous, here you will see the solution is homogeneous. You can't see the different layers, but in heterogeneous you can see the different layers of different substances, right? Next point, let's talk about the size of the solution, colloids and suspension. So in case of solution, the particle size, it varies between 0.01 to 1 nanometer. Okay, so the particles are quite very small, so that's why we get a homogeneous mixture. We can't separate the different particles, we can't identify the different particles, okay, through our naked eyes. In case of colloids, the particle size is 1 to 1000 nanometer approximate. Okay, they are quite large molecules or aggregates, means group of molecules. Now suspension की बात करें तो here the particle size is over 1000 nanometer. Okay, so it is quite more, here the particle size are quite big as compared to colloids, so you can see the particle size through your naked eyes. But in case of colloids, it is very much quite similar to solution, but still the particle size is bigger as compared to solution here. Now solution, if you keep the solution at rest for some time, so you can't see the different layers. For example, if we take the solution of water and sugar and we mix it properly and we leave it for some times, will you able to see the different layers? No. So here the layers or the particles, they do not separate on standing. In case of colloids also, here also the particles do not separate. That's why in the in previously I said that they are quite similar. Like milk is the example of colloid. It is not a solution, okay, because the particle size are quite big as compared to solution particles. Suspension, in case of suspension, if you leave the mixture, so here the particles settle out and you can identify them or you can see it with your naked eyes. Okay, now let's talk about the filtration method. Here the particles cannot be separated by filtration because it is a homogeneous mixture. In colloids also you can't separate the particles through filtration. You have to use other techniques. For example, when you have to take out the butter from the milk, then what you do, you rotate the milk at a very high speed, and this process is known as centrifugation. So here also by filtration you can't separate, but it can be separated by centrifugation method. In case of suspension, the particles, as they are very big in size as compared to these two, so here you can separate it by the filtration method. Now lastly, here the solution, if you pass a beam of light, they do not scatter the beam of light. Okay, so that means they do not show Tyndall effect because the particles are uniformly mixed with each other, so there is no space between them, so no scattering of light is observed, no Tyndall effect. In case of colloids, in case of suspension, you can see the Tyndall effect. The scattering of light is visible, okay, here because the particle size is quite large as compared to solution particles. So we have seen the differences between these three. I hope now it is clear how we can differentiate between colloids, between suspension and solution. So colloids are unlike solution. They are not like solution, as I have already told you, because the particles are much larger than the solution, okay, and they cannot be separated by filtration, but they scatter the light phenomena. So here you can see the similarity also is there, like here the filter, they cannot be separated by filtration. But when we talk about the Tyndall effect, so here in case of colloids the Tyndall effect is seen by the particles, but not in case of solution. So this is all about the differences. Hope you have all have understood. Thank you everyone.